Alright, this is going to be the first recorded lab introduction of the trimester, so we're going to start off looking at the anatomy of the microscope. That's the first thing you'll need to know. So we have these good quality Olympus microscopes. Newer scopes are made of plastic and are more cheaply designed. So these are good scopes with great optics and a nice metal backbone. So these are very good and sturdy scopes. So make sure that you take care of them and treat them properly throughout the course of the trimester. So the different parts of the scope that you need to know are labeled in your lab manual in the beginning, and so I'm going to go over some of the major parts here with a real scope. The main structural components of the scope: you have a flat base here, and then you have a metallic arm. The arm is what you should use to handle the scopes. When you lift the scope, you should grab this part with the arm and then support the base with your other hand, because these are quite heavy, and also that prevents them from being jostled around a lot. And these scopes have filters which can fall out if they're not supported with both hands, so make sure you handle them properly at all times. There is also a headpiece mounted on top of the arm, and mounted in the headpiece are the two ocular lenses. So these eyepieces or ocular lenses can be removed, and in fact you can invert them and look through the other end to use these as simple magnifying glasses. It's called an inverted ocular technique, and we'll actually do a live demonstration of that during a laboratory period. Each of these ocular lenses magnifies the image in the slide by a factor of 10. So this gives you a 10x magnification. This is not the only lens in the scope, however. We'll talk about the other lenses or objective lenses here in a minute. Note that the eyepieces are mounted on this headpiece here, and this headpiece can be fixed in place, or if you loosen this thumb screw below it, you can actually swivel the headpiece, and then you can retighten it to have the eyepieces facing inward 360 degrees, whichever direction you like. So that's kind of a nice feature of these scopes. You'll also note that the interocular distance here can be adjusted. These eyepieces, their width can be adjusted to accommodate the distance between your eyes, because not everyone's eyes are the same distance. So that's kind of a useful feature there. There is a fine focus adjustment on the left eyepiece typically, but not on the right. So that's another thing to look at when you are trying to adjust the image for clarity. The other set of lenses are called objective lenses, and you have different magnification lenses. Most of these scopes are equipped with a basic 4x objective lens, which magnifies the image on the slide by 4 times. There's also a 10x objective, which is longer and magnifies the image by 10 times, and then you have a longer objective still, which magnifies by 40 times. So this would be classified as the 4x, 10x, or 40x objective lenses. Note that these objective lenses are mounted on this rotating turret, this structure right here, which you can use to quickly and smoothly switch objectives without really interfering with the specimen that's placed on the microscope stage. Note that these objective lenses are color-coded on most of the scopes here as well, such that the 4x or lowest power objective has a red stripe on it, the 10x objective has a yellow stripe, and the 40x objective has a blue stripe. Some scopes may have a 100x objective with a white stripe. Most of them do not, however. It's important when putting these microscopes away that they are always stored properly, and proper storage of the scope means that the 4x lowest x objective, the 4x objective should be in place, or if you have a blank objective piece here, you can put that into place as well. You do not want to have the 10x or 40x objective swiveled into position when you store them, because they can make contact with the stage and damage the optics here. So the next part of the scope here, this flat piece, this is what we call the stage. The stage here is actually where you put the sample slide, and the slide there is then clamped into this specimen holder here, and this has a little finger lever here that you can use to pull it back and clamp the slide on in place, and it should be seated firmly on the stage here. Adjustment of the slide across the stage in 2 dimensions is done by adjustment knobs, and by doing that you can move the specimen however you want to on the stage so that you get a clear field of view. Okay, let's take a look underneath the stage, and you can see this structure right here hanging from the bottom of the stage. This is called a condenser, and so in order to view these samples in the scope, it requires that light be shined on the specimen, and so light comes from this illuminator here. This is controlled by a power switch in front of the scope. There is a rheostat on the side of the scope which actually controls the intensity of the illumination, so that ranges from 0 or 1 to 10, I believe, and you can change the illumination to a level that's appropriate for the particular magnification. Typically, at higher levels of magnification, you need to increase the intensity of the illumination. With low magnification, you want to use a lower level of illumination so that doesn't hurt your eyes. So the light will shine up from the illuminator here. There is a filter on most of these scopes, a blue filter that can easily be replaced and sits on top of the bulb here. And the light will pass through this illuminator, through this condenser right here. And the condenser helps to condense the light and focus it directly on the specimen in the slide. Now, this condenser also has an aperture which is controlled by a lever, so by moving this lever you can determine how much light goes into the specimen. There's also another knob here to the side that controls the distance which the condenser is from the stage, and a typical default setting is to have move this knob until the condenser moves all the way up to the stage and then can't move anymore, and then you turn it back a quarter turn back down to the stage, and that should be a setting appropriate for whatever you're going to need to use the scope for the rest of the trimester. Let's take another side view of the scope here, and you can see some other knobs on the side. On each side of the scope you have a large knob right here. This is called the coarse adjustment or coarse focus, and then you have a longer, skinnier knob which is the fine focus adjustment knob. So when you have a specimen on the slide, by rotating the coarse or fine adjustment, it moves the stage up or down relative to the objective lens to help focus the light where it needs to go, and that gives you a better clarity image. If you adjust the coarse focus knob, the stage will move up and down a large amount. That's good for focusing on slides when you initially put a slide on the stage. Once you have it relatively focused, however, and when you're switching to objectives, you simply need to adjust the fine focus knob to switch to another power objective. These scopes are said to be parfocal, which means that you can switch from one objective to another without losing much of the focus, and so you only need to adjust the fine focus knob when switching objectives. But the first time you put a specimen on, you'll probably need to use the coarse focus to get it mostly in focus. So those are the major parts of the microscope that you are responsible for knowing, and that stuff will be on the quiz. This is a section of jejunum, and this particular tissue has been stained with a stain called hematoxylin and eosin. So there are multiple different types of stains we'll use throughout the course of the trimester in this lab. Today you need to know 3 of them. Hematoxylin and eosin we usually just abbreviate that as H&E. So if asked on a quiz, you may feel free to write it out, or you can write it in the shorthand version, just call it H&E. But you do need to be able to know both forms if written to you on a practical exam. H&E is far and away the most common type of stain that is used in this lab for studying human tissues, for doing basic histology, and it renders slides this kind of a reddish purplish color because of the two components within the stain. So H&E is essentially two different stains in one. The hematoxylin component of H&E will bind to acidic structures in the cell because that stain is itself basic, and so acidic structures within the cell have a greater affinity for basic stains, and basic structures within the cell have a greater affinity for acidic stains. So hematoxylin has kind of a purplish coloration to it, and so if you view structures at high magnification, those that take up the hematoxylin have kind of a purplish appearance to them. In most cases, most nuclei in cells will take up the hematoxylin and stain sort of purplish. So you can see the nuclei of these columnar epithelial cells lining the surface of this intestinal villus here. And so all these little purple dots, those are the nuclei of the cells. Nuclei are acidic because they contain nucleic acids, and so because that structure is acidic, it will take up the basic dye hematoxylin. The other stain is eosin, and eosin is an acidic stain, and it binds to basic structures within the cell. The most obvious basic structure within the cell is just most of the cytoplasm outside of the nucleus, and so the cytoplasm of cells typically will take up the eosin and stain it kind of a reddish coloration. Now, structures that are stained with eosin in the cell we refer to as eosinophilic or eosin-loving. However, structures that take up the hematoxylin we refer to as basophilic or basic-loving, probably because saying hematoxylinophilic is kind of a mouthful. It's really hard to say, so we just refer to that as basophilic. So it's important that you become very familiar with those two terms, eosinophilic and basophilic structures, because for most structures, when I'm describing them to you in the lab, I may say this fiber is eosinophilic, and you need to know what that means. Or if I'm saying look at that basophilic structure, it's important that you know what that looks like under the scope. So here the cytoplasm appears eosinophilic because it takes up the eosin, and the nuclei in the cells appear basophilic that take up the hematoxylin portion of the stain. So those are the two components of H&E that you need to be able to know. If I show you a slide that's been stained with that, you also need to be able to just tell me in general what stain has been used to stain that slide. The second stain we're going to look at today is osmium tetroxide. Osmium tetroxide is a heavy metal compound. This is a stain which is generally commonly used to stain lipids. So it is fat soluble, and it will bind to the lipids in myelin sheaths or in adipose tissue, for instance, staining them a dense black coloration. So here you can see adipose tissue where you have these large cells called adipocytes. These cells contain large lipid inclusions that takes up the osmium tetroxide, staining them black. here's what adipose tissue looks like that's not been stained with osmium tetroxide. Rather, it's been stained with H&E. And notice that the lipid droplets are not taking up the stain. In fact, the alcohol wash in the H&E stain ends up removing the adipose, most of the lipids, causing these cells to lose their shape and shrivel up a little bit. So note that this is the same tissue here and here, but stained differently, and so that it has a very different appearance. The final stain we're going to look at is called Wright stain, and Wright stain is a type of Romanovsky type stain. In this lab, we'll just simply refer to it as Wright stain. Wright stain and other Romanovsky type stains are very similar to H&E in their chemistry, but Wright stains has some additional components which make it easier to identify white blood cells and blood smears, which are complex cells that have a complex appearance. And so they will stain structures eosinophilic or basophilic, just like H&E, but they also will stain other components to help differentiate between these different cell types. So right here we can see this cell found in a blood smear. This is a type of white blood cell or leukocyte that we're going to talk about later, called a neutrophil. And you can see its cytoplasm is very chemically neutral. It's not really pink or purple. It doesn't have a great affinity for either of the stains. The nucleus is multi-lobed. There are actually 4 discrete lobes of the nucleus here, and this nucleus, like most nuclei, is basophilic, so it takes up the hematoxylin portion of the stain. But since the cytoplasm is chemically neutral, we refer to this type of cell as a neutrophil. In the upper right here, you can see a different type of cell, which we call an eosinophil, and note that its cytoplasm is very eosinophilic. And down here we have a basophil, which has a basophilic cytoplasm, and for that reason the cytoplasmic granules can be very difficult to distinguish between the nucleus, which is also basophilic. So with this particular stain, you can identify different types of white blood cells that are commonly found in blood smears. So those are the 3 types of stains that you need to know. Once again, the first is H&E. This is what a section looks like, stained with H&E. Here is adipose tissue stained with osmium tetroxide. That's the second stain. And the third one is Wright stain. So if I show you an image of tissue that's been stained with any of these 3 stains, then you need to be able to identify the stain for the purposes of the lab quiz this week. Okay, and then finally we have some electron micrographs of different cells showcasing different organelles and cellular inclusions that you need to be able to identify as well. So let me go over those with you real quick. This is a section of liver, and notice that it has a black and white appearance. That's because electron microscopes do not use light. There's no visible wavelength there. It's using electron beams, and so everything appears in black and white. If you ever see electron micrographs that are colored, the color has been added in later on, so that would be called pseudo-coloring or false coloration. And that's a good way to show certain structures, but in their normal form, electron micrographs are always black and white. So they have a very different appearance from what cellular structures are going to look like when viewed in a light microscope. So this is a hepatocyte from liver, and you can see a large nucleus in the middle of the hepatocyte here, and there are several different types of organelles. We're going to talk about these in a little bit. Most of what you're seeing here, though, are these tubular sac-like structures called smooth endoplasmic reticulum, or smooth ER, and that's all of these structures right here. Now, hepatocytes normally have lots of smooth ER anyway, because smooth ER is involved in detoxification and other functions that liver cells carry out. But these particular cells have been induced to hypertrophy by treatment with a particular drug, which also causes them to express an overabundance of smooth endoplasmic reticulum. So you won't see as much smooth ER in most cells. This cell just has tons and tons of it in this particular part of its cytoplasm. Here's a full view of a nucleus, what a nucleus looks like here. Note that it typically is a round or oval shaped structure, but not always. The appearance of nuclei can vary quite a bit dramatically. But when viewed from an electron micrograph, this is what it looks like. Oops. And you can see that the chromatin is found in a more condensed state, heterochromatin, which is this darker, more electron-dense material. You find it clumped here in the nucleolus region. You can also find it in the periphery of the nucleoplasm. And then you have its less condensed state, or euchromatin, which is the lighter, less electron-dense distribution of the chromatin. The appearance of chromatin and the nuclear envelope, which is a double-layered membrane that encloses the nucleus, should allow you to identify this organelle. Here's a section showing Golgi apparatus. Golgi apparatus has a very layered appearance. It consists of these flat sac-like structures. And these structures appear very much like rough endoplasmic reticulum, which I'll show you in a little bit, but the lumens of these structures are more narrow and they are overall more compressed. Additionally, there is a polarity to Golgi apparatus, meaning that one side is different than the other side. The two sides of Golgi apparatus or Golgi complex are referred to as the cis side, which is the side which faces the nucleus, and then you have the trans side, which is the side facing away from the nucleus. Note that the cis side tends to have a concave curvature, whereas the trans side has more of a convex curvature to it. In this electron micrograph, you can see these structures. They're small, membrane-bound organelles. They're very electron-dense. These are lysosomes. Lysosomes are not normally inherently so electron-dense, but the only real good way to distinguish them from other small organelles or vesicles is to label them with an electron-dense marker that causes them to appear very black. And so that's what these structures are here. They are relatively unremarkable in appearance because they just look like, they may look like other large vesicles in the cell. And so we use electron-dense markers to be able to identify those. You can see two major organellar structures highlighted in this slide. First, let's look at the mitochondrion here. So mitochondria are typically cigar-shaped, kind of elongated organellar structures. They have a double membrane here, and the membrane protrudes internally to form these structures called cristae. In between the cristae, you have the mitochondrial matrix here. I don't know why that keeps popping up here. And so this is what a mitochondrion looks like when viewed in an electron micrograph. Notice in some of these other slides you can see mitochondria as well. Now sometimes they appear more circular in appearance. That's because they're cut in cross-section instead of longitudinal section. So this is what a typical mitochondrion looks like normally when viewed in longitudinal section, but if it's cut in a cross-section, it's going to appear more circular. Sometimes they can look more oddly shaped or asymmetrical, and that's because mitochondria can actually change their shape within the cell. The other major organelle that you see here is called rough endoplasmic reticulum. Rough ER has a very similar appearance to Golgi apparatus, in that it consists of these long, elongated, flattened sacs with a lumen here. But notice that the lumen is larger And rough ER also possesses ribosomes on the surface here, which are these dark electron-dense structures here. And you don't find those with Golgi complex, so they actually do have a very distinct appearance, even though it's common for some students to confuse them. So let me go back a couple slides where you can actually see both structures. This is Golgi apparatus. This is Golgi apparatus here, as well as this. This is rough ER. Notice the studded appearance here due to the ribosomes on the presence of the rough ER, whereas Golgi apparatus looks much smoother. Golgi apparatus looks much more compressed as well. So this is all rough ER. Here's more rough ER. Golgi apparatus, more rough ER here. So make sure that you are able to identify the differences between those two organelles in electron micrograph. Here's another slide with a lot of rough endoplasmic reticulum and lots of mitochondria in various sections. We're going to look at two different cytoskeletal elements, both in longitudinal section and in cross section. So the two major cytoskeletal elements we have here are microfilaments or actin filaments, AF here, and those are these long thin strands that essentially appear to be dark solid fibers. The other type of cytoskeletal element you can see here are microtubules, and microtubules are larger in diameter than microfilaments, and they are hollow, so they actually look like little tubes or straws when viewed in longitudinal section. You can actually see the interior is hollow. In this slide we have glycogen inclusions. Glycogen is found mostly in the liver and also in the skeletal muscle. There are small amounts of glycogen that are stored elsewhere, and they are stored as these electron-dense, kind of granular cellular inclusions, these dark structures that you see here. So be able to identify those. And the second type of cellular inclusion we're going to look at are lipid droplets, and lipid droplets have taken up OSO4 in this case, making them very electron dense. But notice they're really perfect spherical symmetry here. Some lysosomes or other structures that may be tagged with electron dense materials that are circular aren't going to have quite this perfect of a surface symmetry here. So when you get these really nice looking spheres, these are that's indicative of lipid inclusions that you can see here. And so that's everything that we have to show you for lab this week. So things to study for the lab quiz: again, know the anatomy of the microscope, know the 3 different types of stains that we talked about today, and be able to identify the cellular organelles and inclusions shown in this presentation. And that's it.